A very good morning to one and all. This is Mrs. Mary Ratnanita once again, welcoming back to your pharmaceutical microbiology class. So the topic for today's our class is antigen antibody reactions. So in the last class, in the previous classes, we have seen what is immunity and what is immunology, what is immunity, what are the different types of immunity, what are the cells involved in different types of immunity, and what is an antigen, what is an antibody. We have seen the structures of antigen and antibody, how they are formed and how they combine with each other to undergo an antigen antibody reaction. So we have seen all that in your previous classes. So let us see what are the different antigen antibody reactions in your uh, uh, in the present class. So before entering into the topic, let us learn about the learning objectives. So the learning objectives are this, by the end of this topic, the student shall be able to list out the various types of antigen antibody reactions. At the same time, the student shall be able to explain what are the different antigen antibody reactions. Along with that, the student shall be in a position to explain the different applications of these antigen antibody reactions, where exactly you use these antigen antibody reactions, why are they useful to us, where in our day-to-day practical life we are going to use these antigen antibody reactions, which can be explained by the student by the end of this topic. Now entering into your main topic, that is your antigen antibody reaction. So coming to the introduction part, you know what is an antigen and you know what is an antibody. So these antigens and antibodies, they specifically combine with each other. As when I was explaining about the immunity and antigen and antibody, I clearly told you that these antigen and antibodies are specific to each other. So antibodies which are stimulated or which are produced in response to a particular antigen will react with that particular antigen only. They will not react with any other antigen. So that type of interaction between an antigen and antibodies are called as antigen antibody reactions, and they are abbreviated as AGAB reactions. And these reactions, they form the basis to detect various infection causing diseases like your some of the infectious causing disease, disease causing agents, at the same time some of the enzymes which are non-specific antigens. So in order to detect them, these antigen antibody reactions are used. So what are the applications of these antigen antibody reactions? We'll learn at the end of the topic. So when antigen antibody reactions occur in vitro, they are known as serological reactions. So these serological tests involves the interaction between an antigen and antibody uh, in the blood serum. And these are used for the diagnosis of various infectious diseases like autoimmune diseases, and also for the grouping of the blood, human blood and animal blood. So you know we humans have different blood groups. So just to find out what blood group you possess or what is our blood group, even for a grouping purpose, we use these antigen antibody reactions only. And also in order to test certain tissues prior to the organ transplantation or any transplantations, during that time also we go for these antigen antibody reactions. So when we are talking about antigen antibody reactions, when we have learned about the structures of antigen antibody, I clearly told you that antigen has a specific chemical portion to which the antibody binds, and that portion is called as epitope. So the antigen associates with antibodies at the epitope, that is nothing but antigen determinant site, which is present on the antigen, and this site will be bound by the complementary region which is present on the antibody. So what are we saying here? The antigen possesses a chemical region, a portion of chemical region, which is called as epitope or antigen determinant site, which is present on the antigen. And this region would be complementary to the complementary determining region on the antibody. So if they are complementary, then the antibody will come and bind with the epitope of the antigen, and this binding will be done by various non-covalent forces, which are called like your hydrogen bonds, your van der Waals forces, your hydrophobic forces, your electrophobic bonds, and different electrostatic bonds and all. So the here a large number of covalent bonds are required to form a strong bond between an antigen and antibody. So the antigen antibody reactions are very specific in nature. So as I said, since antigen is specific to antibody and antibody is specific to antigen, they are very specific in nature. And upon exposure to specific antigen, the antibodies interact with that particular antigen and form an antigenic antibody complex. So binding is facilitated by the antigen determinant site, and this binding is done by a strong bond. The bond which is formed between antigen and antibody would be very strong, and that is called as the strength of the such bond is called as affinity. So when the antigen antibody bonds are not formed properly, you cannot have the antigen antibody reactions, and these reactions are useful for many purposes. We'll see what are the uses of these antigen antibody reactions and applications of antigen antibody reactions by the end of this topic. Since the body is constantly exposed to different viruses, different antigens, these antigen antibody reactions serve a very vital and important role in your immune as an immunological barriers. So what is happening here? See, and your microorganisms and antigens are present everywhere. They're ubiquitous. You cannot say that this is the area where there are no microorganisms, there is no antigen present. So since our body is getting affected by antigens in all ways, in every ways, in from food, from water, from the air, from by all means, so these antigen antibody reactions they serves as uh, they play an important role as immunological barriers. And apart from that, they have many importance, like they are used in diagnostic purposes. We'll see the application part. Now, when you talk about the antigen antibody reactions, so these antigen antibody reactions occurs in three different stages. So in the first stage, the reaction involves formation of antigen antibody complex, which we were discussing till now. And in the second stage, it leads to visible events like precipitation and agglutination. And in the third stage, the includes the destruction of the antigen or its neutralization. So the first stage, antigen and antibody will combine. Second stage, precipitation or agglutination takes place. Third stage, destruction of the antigen takes place, or neutralization of the antigen takes place. We cannot simply say that definitely it gets destroyed. It sometimes gets neutralized also. So now talking about the various antigen antibody reactions. When we are talking about the various antigen antibody reactions, there are different types of antigen antibody reactions, and they are precipitation reaction, when agglutination reaction, neutralization reaction, complement fixation reaction, ELISA, that is nothing but enzyme linked immunosorbent assay, and FAR, that is fluorescent antigen reaction. So we are going to see one by one what are these reactions and how the antigen reacts with antibody. Now talking about your first reaction, that is precipitation reaction. So in precipitation, generally the antigen has more than two sites to which the antibody has more than two sites which binds with the antigen, and antigen has more than one site to which the antibody bounds. So generally, when antigen and antibody in proper concentration, when they're bound, they form insoluble uh, lattice, which eventually turns into a visible precipitate. So when a soluble antigen, when combines with an antibody in the presence of an electrolyte like your sodium chloride, at a particular temperature and pH, definitely, it forms an insoluble precipitate of antigen antibody complex. So what are we saying? When the soluble antigen, when it combines with the antibody in the presence of electrolyte and temperature and pH has to be maintained, it forms an insoluble complex of antigen antibody precipitate. So the antibody that is causing precipitate is called as precipitate, and the reaction is called as precipitation reaction. Since here the precipitation is formed, we are calling it as precipitation reaction. Now, when we talk about this is how the precipitation reaction goes on. So what you have to do is, generally this precipitation reaction takes place in two different steps. The first step is rapid, which occurs in few seconds. And here in the first step, it leads to the formation of antigen antibody complex. And the second step is a bit slower when compared to your first step. It takes from few minutes to few hours. And here it leads to the formation of insoluble lattice, which eventually turns into a precipitate. So here what you do is you have to take the different test tubes, a series of test tubes, and you have to add the constant amount of antibody in all the test tubes. And after that, you have to add your antigen in increasing amounts. Like if you add 1 ml in the first test tube, you have to add 2 ml, then 4 ml, then 8 ml, then 16. So it has to be an increased pattern. So what are you doing? You're taking a series of test tubes, adding antigen, constant amount of antibody, and to this you're adding your antigens in increasing amount. Then after that, what you have to do is you have to allow the keep the test tubes aside and allow the antigen to react with the antibody and uh, allow the precipitate to form. And once the precipitate is formed, you have to take the tubes and subject them to centrifugation. And after centrifugation, you have to decant the supernatant liquid, and whatever the precipitate which is formed, it will settle down as a pellet. You have to take out the pellet, collect the pellet, and weigh it. Then you have to draw a graph taking uh, the um, uh, antibody precipitate, what the quantity of the precipitates on an increasing amount of antigen on. So you have to draw a graph taking increasing amount of antigen on x-axis and precipitate on the y-axis, and then you have to draw a graph. Now you see that if at all the antigen and antibody concentrations are more, they are not appropriate, the precipitate form will not be apt. So the region of zone of equivalence is characterized by optimal levels of your antigen and antibody, that is maximum precipitation will be seen. So there you can say that particular is the maximum zone of. So you'll have three zones: zone of antibody, zone of equivalence, and zone of antigen. So you are considered about your zone of equivalence. That is, this is considered as the apt zone for your checking your antigen antibody reactions. So generally this precipitation reaction takes place in liquids as well as in gels. So this is what we have discussed just now is a liquid precipitation part. And precipitation in gels, what they do is generally they take the microtiter plates and they make wells and slots in the gel. Wells and slots means they dig the holes and straight slots like in the gel. And alternatively, they put antigen and antibody. And then after that, they run the electrophoresis. So this electrophoresis will help in separating the proteins according to its concentration gradient. So what happens, these proteins will run, the antigen and antibody will get spread over the gel, and at one concentration, the antigen runs towards the antibody to which it is complementary. And you can see a precipitation arch that is considered as your required precipitation arch. So that is required, that is considered as a required concentration. So that is about your precipitation reaction. Now talking about your next reaction, that is agglutination reaction. So agglutination, the name itself says agglutination, aggluting, forming, clots, clumping, uh, uh, joining together. So when a particular antigen is mixed with its antibody in the presence of an electrolyte at suitable temperature and pH, the particles are clumped or agglutinated. That means they join together, they form clots or clumps. So the antibody of the serum causes antigens to form clumps, and that antibody is called as agglutinin. And the antigen that is undergoing agglutination is called as agglutinogen. So what are we saying? The antibody is responsible for causing the antigen to form clots or clumps. And that antibody is called as agglutinin. And the antigen which is undergoing agglutination and forming clots is called as agglutinogen. And if at all these agglutination reactions, you have different types of agglutination reaction. And if at all blood is involved in this agglutination reaction, we call it as hemagglutination reactions. So what are the different types of agglutination reactions? We'll see. There are different types of agglutination reactions. They are slide agglutination, they are tube agglutination, passive agglutination, and agglutination inhibition. So when you talk about slide agglutination, this is a rapid method in order to determine the presence of an agglutining antibody. So what you have to do is you have to take a slide and you have to add uniform suspension of the antigen. And to this, you have to add a drop of saline. And to this, you have to add a drop of antiserum, uh, which contains antibodies. So, and after that, you have to take a thin loop or a small glass rod and just need to mix the antigen with the antiserum. And after that, wait for some time. If at all the antigen is specific towards the antibody, or antibody is specific towards the antigen, then what is they will form clots or clumping, or agglutination is seen, and coagulation of the antigens will be seen. So those are considered as positive one. So for example, the generally slide agglutination method is used for your blood grouping type as you have already practiced in your anatomy, how you go for your blood grouping, you take the antigen, you add the antiserum, and then you check it. So that is where this slide agglutination is applicable. Then talking about your tube agglutination. So generally this tube agglutination is the standard method of qualitative estimation of the antibody. So what you do is serum that is containing antibody is diluted serially with saline in several small test tubes. So you have to take test tubes, different number of test tubes, and then you have to add the serum that is containing antibody along with the saline. It has to be diluted with the saline, then that has to be added. And to this, constant amount of antigen is added. So what are you doing? You're taking the antibody, you're diluting it with the saline, and then you're adding it into the test tubes. And to this, constant amount of antigen is added. In precipitation reaction, what we did, we took constant amount of antibody and increasing amount of antigen is taken. Here, antibodies are diluted and antigens are added at constant rate. And a control is also kept, which does not contain any antiserum. Then the tubes are incubated until the visible agglutination is formed or is observed. And then these agglutination, whichever shows the agglutination, considered as positive. So generally, this type of agglutination, tube agglutination test is used in diagnosing typhoid, your typhus fever, so to perform viral test, in order to perform brucellosis. So all to perform all these are the applications of your tube agglutination. Talking about your passive agglutination. So passive agglutination here similar to the hemagglutination, but physical nature is altered here. So generally, antigens is coated on the surface of a carrier particle, which is called as a carrier particle, and they then they convert precipitation reaction into agglutination reaction. So what are we doing here? We are converting the precipitation reaction into agglutination reaction by coating the antigen with certain compounds. So generally carrier particles used here are either RBCs or latex or bentonite. So these are the substances which are used. So we are coating the antigen with these substances, and then we are adding the antibody, and then we are trying to see whether agglutination occurs or not. So when generally patient serum is mixed with these, it leads to agglutination. So the application part, generally in order to diagnose the rheumatoid arthritis, in order to get the rheumatoid arthritis test done, we go for your passive agglutination. Now, last comes is your agglutination inhibition. So, in all these tests, the above methods, slide agglutination, tube agglutination, passive agglutination, we have seen that here we are confirming the test as positive if at all the agglutination is seen. But here in the last test, we confirm the test to be positive if at all the agglutination is not seen. That's why it is called as agglutination inhibition reaction. So generally what happens in agglutination reaction, inhibition reaction is, so that's generally it is a very highly sensitive method and can detect even small amounts of antigen present also. So in order to find out the pregnancy positive or negative, we generally go for this agglutination inhibition test. So what happened nowadays, we are getting ready-made pregnancy testing kits. So these pregnancy testing kits contains latex particles which are coated with human chorionic gonadotrophic hormone, that is HCG hormone, and this HCG hormone will be present only in the urine of the woman who is pregnant. So normal people will not have this, the urine of the normal woman will not have this hormone. Only the woman who is pregnant will have this hormone in her urine. So what are we doing? We are taking the kit which is coated with the latex particles containing your HCG hormone and anti-HCG antibodies. So after this, what you do is you have to add an anti-HCG carrier conjugate to this mm, latex particles. So what is happening? So as I said, only the urine of a pregnant woman will have this HCG hormone. So you have to take the urine sample of the pregnant the woman who is suspected to be pregnant, and it is the sample is incubated with anti-HCG and carrier conjugates for suitable time and suitable period of time. If at all, if HCG is present in the urine, that means if at all the woman is pregnant, the HCG will go and bind with the anti-HCG antibodies. and the carrier conjugates will be free. So that's why there won't be any formation of agglutination or clotting. If at all the urine does not contain HCG, so these carrier conjugates will go and bind with the anti-HCG antibodies, and they forms clots or clumping or agglutination. So, so if at all the HCG is present, it will not allow the carrier conjugate to bind with the HCG antibodies. So thereby there won't be any agglutination formation. If at all the woman is not pregnant, then the carrier conjugates will have the chance to go and bind with the anti-HCG antibodies. Thereby, the clotting will be seen, and it is considered as negative. So here we are coming to a conclusion. We are showing the results as positive or negative. If there is no agglutination, then the result is positive. If agglutination is there, the result is negative. Hence, the name is termed as agglutination inhibition reaction. So that is about your agglutination reaction. Next is your neutralization reaction. So generally, reaction involves blockage of toxic effects of certain microorganisms or virus or pathogens or antigens by formation of specific antibodies in the host. So generally, those substances capable of neutralizing bacterial toxins are called as antitoxins. So how they how they neutralize the effect of toxins? So these antitoxins, they go and bind with the antibodies, and they will not allow the toxins to go and bind to the target site. Thereby, they neutralizes the activity of the toxins. So hence they are called as, they form a toxin antibody complex and will not give the chance for the toxins to go and bind with the target site and neutralize the activity. So generally this kind of uh, neutralization reactions can be seen in diagnosing diphtheria or for using tetanus and all. These are the application parts. Then coming to your next test, that is complement fixation reaction. So the name itself says complement fixation. So what are we doing here? Generally, the phagocytes are capable of attacking the invading antigens or microorganisms and undergo phagocytosis. But if this process of phagocytosis can be rendered efficiently by uh, the another process which is called as absonization. That is, absonin is nothing but a complement. It is nothing but a specific component that is complement to your antigen. So the lysis of the RBC or bacteria requires non-specific unstable components of fresh serum, which are called as complements. So what you do here is, this complement system generally comprises of 11 proteins and almost all present in all the individuals. So these complements will bind to the Fc component of the antibody involved in antigen antibody reactions. So when we were dealing with the antibody structure, I told you that there would be Fc and Fab portion. So these complements will go and bind with the Fc portion of your antibody, which is involved in antigen antibody complex formation. And this ability to of antigen and antibody complex to fix the complement is called as complement fixation. Generally, it is used in complement fixation test. So in this test, first step, what you do is you have to add the antigen, take the antigen and antiserum, and it has to be heated up to 56 degrees in order to inactivate the complement. And then they are mixed with the known amount of your complement. First, what are you doing? You're taking the antigen, that is, you're taking the serum, and you're heating it. And once you heat it to 56 degrees, whatever the complements are present with, they'll be inactivated. Then you have to add the known concentration of the complement. And after adding that, you have to incubate this for 18 hours at 4 degrees. Then if antibody is specific to the antigen is present in the serum, then antigen antibody complex will be formed. And this will fix the complement. So hence it is called as complement fixation reaction. So that is about your complement fixation reaction. Generally, so next is your ELISA. So ELISA is nothing but enzyme-linked immunosorbent assay. You might have learned about this ELISA in your previous classes. Let's just have a brush up about this reaction. So in 1971, so enzyme-labeled antigens and antibodies were developed as a serological agents for the assays of antibodies and antigens. Assay is nothing but quantitative and qualitative measurement of the particular protein in a particular given sample. So in order to find out whether the antibodies or antigens are present in the given sample, and if at all the proteins are present, then what is the quantity of this protein? We perform this assays. So these ELISA are very simple, they are sensitive, they are economic, and when you compare to your radioimmune assays, they are less hazardous. And the ligand used here is a molecule that can detect the antibody and is covalently coupled with an enzyme, which such as your peroxidase, beta galactosidase, alkaline phosphatase. So here what happens is in ELISA, so the name itself says enzyme-linked immunosorbent assay. So it is also called as uh, enzyme immunoassay. So what you do here is, here we use, generally in antigen antibody reactions, we are using one antigen and one antibody. But in ELISA, we use two antibodies, one primary antibody, one secondary antibody. The primary antibody is the one which is specific towards the antigen to form the antigen antibody complex. The secondary antibody is used to link the enzyme. Hence, the name enzyme-linked immunosorbent assay. And generally, these enzymes would be your peroxidase, your beta galactosidase, your alkaline phosphatase. These are the enzymes which are generally linked and which carry on the reaction. And these enzymes are catalyzed by colored substrates, and they show the result in the formation of color change. So that is about the introduction part of your ELISA. And when you come to the talk about the different types of ELISA, you have three different types of ELISA: sandwich ELISA, indirect ELISA, and competitive ELISA. So when you talk about sandwich ELISA, so generally what you do here is you have to take your microtiter plate, and this microtiter plate has to be coated with purified antibodies uh, uh, which are specific to the antigen. Then uh, the sample containing the antigen has to be taken. They have to be added to the microtiter plates. And then so generally these sandwich ELISA, as I said, so microtiter plates are taken. You have to coat with antibodies which are specific to antigen. And then the serum containing the antigens will be added to the antibodies. And then they're allowed to react, and you have to wash the microtiter plates in order to remove the unbound antigens. And then what you have to do once the washing is completed, this has to be washed with a suitable buffer at suitable pH. And after the washing is completed, you have to add the secondary antibody linked with enzyme, and this antibody should link with another epitope of the particular antigen. So whatever the antigen is present, to one epitope, this antibody would be bound. So antigen antibody complex is formed. So you are adding another antibody which is complementary to the another epitope of that particular antigen, and then you are adding the you're linking the enzyme with that antibody, and you're adding it. So after that, you have to add suitable substrate which is colored, and then you have to allow the reaction to happen. The substrate is catalyzed by the enzyme is catalyzed by the substrate, and you can see the color change in the reaction. So the intensity of the color is directly proportional to the concentration of antigen in the serum. So what are you doing in your sandwich ELISA? You're taking the microtiter plates, you're coating it with the antibody which are specific towards the antigen. Then you're taking the sample containing the antigen. Then you're adding this sample to the microtiter plates. Then you're allowing the antigens and antibodies to react and to form a complex, washing off the excess of antigens which are not bound. Then you have to add secondary antibody which are linking the enzyme, containing the enzyme. And this antibody will bind to another epitope of this antigen. And then what is happening? You have antigen, antibody, antigen, and above that you are adding another antibody. So hence the name sandwich ELISA. And after that, you're adding the suitable substrate, the substrate suitable to the enzyme, and then the enzyme is catalyzed by the substrate, and color change is same, and intensity of color is directly proportional to the amount of antigen present in the serum. So that is about your sandwich ELISA. Now talking about the indirect ELISA. So in indirect, this technique is used for detecting generally HIV, and the envelope proteins are developed by recombinant DNA technology and coated on the surface of the microtiter plates. So here what you do is, uh, you take the microtiter plates, you add the plates are coated with antigen, and then they are added with non-reacting proteins like your bovine, your casein, etc. Then the biological sample containing the antibodies are added to it. First, what you're doing, you're taking the microtiter plates, adding the antigens. Then non-reacting proteins are added. Then to this, the biological samples containing the antibodies are added. Then you're allowing the antigen and antibody to react. And then after the antigen antibody reacts, the complex is formed. The unbound antigens antibodies are washed off. Then you're adding the secondary. Process is same as that of your sandwich. Secondary antibody is added, and then your uh, um, washing of the unbound antibodies, and then substrate is added, and then color changes seen, and intensity is directly proportional to your antigen present. So that is about your indirect ELISA. Now talking about your competitive ELISA. So competitive ELISA is nothing but it's another variation for measuring the amounts of antigen in the present in the given sample. Hence, it is called as competitive ELISA. So in this technique, so first what you do is you take the unlabeled primary antibody. So first you take the antibody, and then you incubate this antibody with the serum sample containing antigens, and you allow the antigen and antibody complex to form. Then what you do, you take the microtiter plates, and to this microtiter plates, you are adding antigens. And after antigens are added, you're adding your antigen antibody complex which has been formed. And then, so the more number of antigen present in the sample, the less free antibodies will be available for antigen to bind. And then the secondary antibody is added linked with enzyme, and then substrate is added, and the procedure is same. So what you're doing here, first you're taking antibody, then you're taking the biological sample containing the antigen, you're adding them, you're incubating them, and allowing the antigen antibody complex to form. Then you're taking the microtiter plate, you're coating it with the antigens, and then you're adding this antigen antibody complex to this microtiter plates, washing off the unbound antigens and antibodies, and then you're adding your secondary antibody, which is linked with enzyme, and then you're adding your substrate, allowing the reaction to happen. Depending upon the color change, intensity shows the amount of antigen present in the sample. So this is how generally we go for your competitive ELISA. So in sandwich ELISA, first you're coating the microtiter plates with antibodies, then you're adding antigens, and again secondary antibodies added. In indirect ELISA, you're taking antigens on the microtiter plates, you're adding antibodies from the biological serum, and then secondary antibody is added, then you carry on the test. In competitive ELISA, you're taking antigen antibody complex. First, initially itself, you're making the complex to form, and then you're taking the microtiter plates, adding the antigen, adding antigen antibody complex to this microtiter plates containing antigens. The antigens which are as the number of antigens are more in the sample, then the number of antibodies which are available to bind for the antigens will be less. So after that, you have to add the secondary antibody and then go with the procedure. So this is what your ELISA is about. So this ELISA is generally used to diagnose. It is used as a diagnostic tool in most of the medical sciences, in medicine and also in plant pathology. And it is also a quality control check in various industries. So this is used in various methods and various ways. So that is about your ELISA. Now talking about your last, that is your PAR, fluorescent antigen reaction. The name, as the name itself, the technique utilizes fluorescent dyes like your fluorescein, rhodamine, your phycoerythrin. These are the fluorescent dyes. So generally what you do is these dyes are tagged with the specific antibodies to be determined in the sample. So what you do is, as the name suggests, the fluorescent dyes are involved in it. The results will be seen as fluorescence. And these type of reactions are, have advantages like they are sensitive, they are specific, and the results are very instant. And these fluorescent dyes, FARs are again of two types: direct FAR and indirect FAR. In direct FAR, generally direct FAR helps in detecting the invading microorganisms or pathogens in the clinical samples. So what you do, specimens, what you do is, in this you have to take a clean slide. You have to add the specific, you have to coat the slide with the antigen. Then specific antibody is conjugated with the fluorescent dye, and then that is added. If at all the antibody is complementary to the antigen, they form a complex, and that can be seen under the microscope, fluorescent microscope, as a fluorescence. So that is your direct PAR. When you talk about indirect PAR, what you do is here, this indirect PAR is usually used in detecting the antibodies in the serum after infection. So during the infection, we are using direct PAR, and after the infection occurs, we are using your indirect PAR. So here also what you do is you take a clean slide, you add your antigen to this, and then you add antibody which is specific to the, you have to link the, which is specific to the antigen is added, and then you allow the antigen antibody reaction to occur. Then after that, you have to add secondary antibody, another antibody which is called as anti-human immune serum globulin. Anti-human immune serum globulin. And this antibody is conjugated with your fluorescent dye. And when you add this antibody to this, if at all the antigens are complementary to the antibody, if the complex is formed, then only this dye will be taken. Otherwise, this dye will not be taken. So when you observe it under microscope, if at all the antigens are complementary to the antibody, they will observe as, you can see them as fluorescence under the fluorescent microscope, otherwise not. So that is about your indirect PAR. So these are the different antigen antibody reactions generally you we go through. And coming to the application part, so these antigen antibody reactions are generally useful in detecting the blood groups for transfusions. So before if a person needs any blood in emergency cases, so you just cannot randomly give any blood to that person. You have to give only that blood group which is complementary, which a similar blood group can be given to the patient. So in order to find out the blood group, whether it is similar or not, we go for blood group, this antigen antibody reactions. Along with that, you have serological exposure for the infection. To identify the infectious agents also, you go for your blood tests using your antigen antibody reactions, and development of immune assays for the quantification of various substances, like whether the particular antigen is present in the given sample or a particular antibody is present in the given sample. So if at all they are present, what is the quantity of that particular antigen or antibody in the particular sample can be identified by using your antigen antibody reactions. And to detect the presence or absence of proteins in the serum, and also in determining the characteristics of certain immune deficiency diseases. So in order to identify, diagnose certain diseases also, you use the antigen antibody reactions. So these are the different antigen antibody reactions and their applications. This is very, very important topic from your examination point of view. Hope you all understood the topic. You can get this topic from NKJN, or you have this topic in JQB, and you have a lot of information in your internet. So feel free, go through it. Don't skip this topic. Very, very important. Almost, you can say, one of the mostly asked, frequently and asked question. So if you have any doubts, feel free to ask me anytime, anyway. Am I available? So you can call me, contact me, message me, and get your queries clarified. So with this, we have come to the end of this topic. Let's meet with a new topic in the next class. Thank you.